Hello everyone and welcome to today's The Scientist webinar. I'm Catherine Lloydell, Scientific Technical Editor for The Scientist, and I will be moderating our discussion. Today our speakers Dr. Matthias Stadtfeld and Dr. Kejin, who will be discussing cellular reprogramming and the next steps in this fascinating field. We like our webinars to be interactive. We encourage you to send us your questions or comments at any point during the webinar, and the speakers will address these during the Q&A session following the presentation. To ask a question, simply click on the Ask a Question tab and type your query into the question box located on the bottom left of your screen. We'll try to address as many of these questions as we can during our Q&A session. Our webinar platform is user-friendly. You can expand the presentation window by simply clicking on the diametrically opposed arrows in the upper right-hand corner. This will maximize the display within your screen. The webinar will be archived on the Scientist website, and we will send you the link via email within a couple of days. Please note that you will not be able to download the presentation slides. I'd like to take this opportunity to thank our webinar sponsors. Bio-Techne brings together the prestigious life science research brands of R&D Systems, Novus Biologicals, Tocris Bioscience, Protein Simple, and Advanced Cell Diagnostics to provide researchers with high-quality reagents and instruments. We specialize in high-quality antibodies, proteins, immunoassays, small molecules, RNA probes, and instruments for protein and genomic characterization. NanoString. Accelerate your stem cell research with NanoString's Encounter PlexSet gene expression reagents. Choose from pre-selected pathways such as stem cells, stem cell signaling, and stem cell transcription factors, or customize a panel up to 96 targets. PlexSet enables you to run 96 samples at a time, generating up to 9,216 data points per run, with only 30 minutes of hands-on time. Axol Bioscience, the human cell culture specialist, provide highly validated iPSC-derived cells together with optimized media and growth supplements. Through the supply of differentiated cells derived from healthy donors and patients of specific disease backgrounds, Axol aims to provide tools for building the best human cell-based disease models and assays to advance biomedical research and drug discovery. Axol human iPSC-derived cell products include cerebral cortical neural stem cells, sensory neurons, microglial cells, astrocytes, motor neurons, dopaminergic neurons, ventricular cardiomyocytes and atrial cardiomyocytes, renal proximal tubular cells, endothelial colony-forming cells, retinal pigment epithelial cells, and melanocytes and sebocytes. Axol iPSC-related services include cell reprogram with Sendai virus or episomal vectors, iPSC gene editing, and iPSC differentiation to target cell types. Combining our passion for science with quality, innovation, and customer service, we deliver the robust and reliable products our customers need to advance their research faster. Stem Cell Technologies is a biotechnology company that develops specialty cell culture media and cell separation products to support research in the fields of immunology, hematology, neuroscience, mesenchymal cell, stem cell, and epithelial cell biology. We also provide instruments, cytokines, antibodies, training courses, proficiency testing, and contract assay services. Driven by science and a passion for quality, Stem Cell delivers over 2,500 products to more than 80 countries worldwide. Stem Cell Technologies provides consistent, unfailing quality and one-on-one assistance to help you succeed. Our sponsors have provided us with some helpful resources related to stem cells. We have posted these in our resource list located on the left side of your screen. You can access and download these documents at any time during the webinar. And with that, let me introduce our first speaker, Matthias Stadtfeld. Matthias Stadtfeld studied cell biology in Osnabrück and Heidelberg, Germany, before joining the laboratory of Thomas Graf at the Albert Einstein College of Medicine in New York. During his PhD work there, he investigated the developmental plasticity of blood stem cells during mouse development. As a postdoc with Konrad Hockeblinger at the Harvard Stem Cell Institute in Boston, he made early contributions to the burgeoning field of induced pluripotency. Since 2012, he has been running his own lab at the Skirball Institute at NYU Langone Health, focusing on understanding the epigenetic control of physiological and pathological cell fate changes. Dr. Stadtfeld?
Yeah, hello. Thank you very much for the introduction, and thanks to everyone who tuned into this webinar. So My group's interested in basic molecular properties of pluripotent stem cells. And I'm going to talk about two aspects of pluripotent cells that we consider relevant for their biomedical use. The first one is the occurrence of detrimental epigenetic abnormalities in pluripotent cells. And the second one are molecular and functional features that distinguish different pluripotent cell states. What I'm going to do, I'm going to start with a rather general background to make sure everybody is on the same page, in which I will talk about stem cells, how reprogramming can be achieved, and what is really the therapeutic potential and specific roadblocks for the realization of the therapeutic potential of pluripotent cells. Then I will switch over to present some aspects of our own research, which, as I said, is really using reprogramming technology as a study tool to investigate basic mechanisms of epigenetic gene control. In the first part, I will focus on the dysregulation of imprinted genes in pluripotent cells, and in the second part, I will talk a little bit about what we learned from a highly efficient reprogramming system we developed with respect to the acquisition of developmental pluripotency during cellular reprogramming. So to call yourself a stem cell, you have to really be able to do two things. You have to be able to self-renew, to make more of yourself, and you have to be able to give rise to differentiated cell types. And what distinguishes now different cell types is are the intrinsic and extrinsic factor that balance self-renewal and differentiation, and of course the cell type that a stem cell can generate. And broadly, we distinguish between two types of stem cells. The first are adult or multipotent stem cells, and they are found in many of our adult tissues. Their potential is restricted to giving rise to cells that correspond to their tissue of origin. For example, the best probably studied of these stem cells are blood-forming or hematopoietic stem cells, and they give rise to only blood cells. Adult stem cells have lifelong activity within the body, but in culture, unfortunately, most of them are rather short-lived. On the other hand, there are embryonic type or pluripotent cells, and those can be derived from early embryos, but also from germ cells or via reprogramming, as we shall see. Their potential is very broad. They can give rise to all adult cell types and germ cells. Examples are embryonic stem cells and induced pluripotent stem cells. In the body, the state of pluripotency is really very transient, very short-lived. It's only there around implantation. But in culture, we can grow these pluripotent cells essentially forever. And this is shown here. From a single pluripotent cell, you can generate really billions and billions of identical pluripotent cells. And that, of course, allows you to do really large-scale experiment, biochemistry experiment, large-scale differentiation experiment. It also affords you with the opportunity to do genetic engineering. You can fix defects, you can introduce new mutations, and so on. And in addition to self-renewal, these cells have the ability to respond to developmental signals and differentiate into all adult cell types. And in mice, this can be beautifully demonstrated by injecting pluripotent cells back into early-stage embryos. So that's shown here. GFP-labeled pluripotent cells injected into blastocysts give rise to mice which are entirely derived from these injected cells. So this is, of course, a beautiful and very powerful assay, and the existence of such assays is one of the reasons why my laboratory is working largely with mouse pluripotent cells. So how do we derive pluripotent cells? The gold standard are embryonic stem cells, which can be derived from early blastocyst stage embryos of different species by explant culture. So it is the inner cell mass of the embryo that then gives rise to embryonic stem cells. Embryonic stem cells have been extremely powerful tools for basic research and also for biomedicine, but of course they entail the use of embryos, which can be legally and ethically challenging. And by their very nature, an ES cell line can of course never be fully matched to a given patient. So the alternative then approach to establish pluripotency is reprogramming an adult cell so that it adopts features of pluripotent cells. That can be achieved by cell fusion, so we take a somatic cell, fuse it with a pre-existing pluripotent cell line, and we end up with pluripotent hybrid cells. We can also do this by nuclear transfer or cloning. Here we remove the nucleus from an oocyte and replace it with a somatic cell nucleus, yielding so-called nuclear transfer-derived embryonic stem cells. Both of these techniques are very powerful, very informative. They have their shortcomings, as they are listed here on the bottom, but what they showed very clearly that there must be soluble factors within ES cells within oocytes that can reprogram this somatic cell genome. And that was really the major contribution from Yamanaka and Takahashi, that they identified a defined set of such factors that were introduced in skin cells could convert them into pluripotent cells. So they found that expression of embryonic associated transcription factors OCT4, SOX2, KLF4, and MYC established pluripotency in skin cells, giving rise to so-called induced pluripotent stem cells. And iPS cell reprogramming is, of course, very straightforward. It's very tractable. You can really study what a factor is doing during this process, and it requires no embryos. iPS cell reprogramming is also universal. You can take many different somatic cell types as your starting cell. You can isolate them from different mammalian species, but the state of pluripotency that you are establishing is species specific. So rodent iPS cells, but also rodent embryonic stem cells resemble the pre-implantation embryo, which we refer to as naive pluripotency. While primate ES cells and primate iPS cells, including human, resemble the post-implantation embryo, a stage that we refer to as prime pluripotency. So that's a nice academic difference, but not only that. So first of all, of course, we have different transcriptional regulators that govern naive and prime pluripotency. And I'm going to get back to that a little bit in the second part of my talk. But there are also practical differences. In general, it is much easier to work with and to manipulate naive pluripotent cells. They grow well at clonal density. Genetic engineering is rather straightforward, even though with the advent of CRISPR-Cas, this is not so much of an issue anymore. They also, naive cells, incorporate very well into pre-implantation embryos, while prime pluripotent cells do not. But coming back to the big picture potential of reprogramming technology, the hope is, of course, to fix genetic defects in patient-specific stem cell lines, then differentiate them into the affected tissue in the patient, transplant them back in cell therapy, and cure the patient. So this is attractive. This is also very optimistic. What's flying a little bit under the radar, but is very actively ongoing, is the use of patient-specific stem cells for disease modeling and drug screening. And of course, here, if you find a single drug, you don't have one patient, you have thousands of patients. So that is the promise, but there's of course also there are also of course issues. We have become pretty good at reprogramming, but reprogramming nevertheless might fail. In particular, if we start with maybe suboptimal starting cells. It might also be incomplete, so our attempts of erasing somatic cell identity might be not fully successful. Reprogramming can introduce different abnormalities, coding mutations, epigenetic abnormalities, and so on. And even though we have conditions that allow us to grow pluripotent cells very well, we also have realized that many of these conditions actually over time favor the introduction of additional abnormalities into cells. And this is in particular problematic in the context of human cells, because there we lack stringent assays such as the blastocyst injection that I showed you before to really evaluate what is a good cell line, what is a bad cell line. And finally, an issue that I'm not going to talk about much except for mentioning it here is that our attempts or our protocols of in vitro differentiation in general yield cells that are of lower quality than what we find in our body. So having said all of this, we conclude that even though we have learned much about reprogramming and pluripotency, there's still more to be learned before we can really fully efficiently make the most out of these cells. Now I'm going to talk about two aspects of our work. Again, we are basic biologists, and the first addresses the issue of stability of genomic imprinting in pluripotent cells. So imprinting is really a paradigmatic regulatory mechanism operational in mammals, and it's established by DNA methylation marks that are laid down in germ cells in a gender-specific like manner. And this is shown here in this example. You can see in the upper illustration that the methylated imprinting control region of the paternal allele of this particular gene cluster allows expression of gene A but represses gene B. And on the maternal allele now, the absence of methylation allows expression of gene B but represses gene A. So imprinting establishes an asymmetry in gene expression. And there are about 100 mammalian genes that are regulated in this manner. Many of them are potent regulators of fetal growth, and consequently dysregulation of imprinting, which is also known as loss of imprinting, is a common cause of developmental syndromes, but also predisposes to cancer and other diseases in the adult. And imprinting has been observed in several instances in pluripotent cell cultures. So how did we become interested in imprinting? So we asked a very basic question a couple of years back now. Are there intrinsic differences in ES cells coming from blastocyst stage embryos and in iPS cells coming from fibroblasts by reprogramming factor expression? A straightforward question, but to address this thoroughly, we needed to generate some tools. And the key tool here really was using gene targeting to generate a doxycycline-inducible allele that encodes from a polycystronic cassette all four of the Yamanaka factors, OCT4, KLF4, SOX, and MYC. If you're familiar with human iPS cells, then you might recognize that this cassette is identical to the stem cell cassette that was originally described from Gustavo Mostoslavsky at Boston University. So what did we do with this? With these gene-targeted ES cells now, we generated via blastocyst injections and somatic cell isolation and reprogramming a set of completely genetically matched ES cells and iPS cells that we then compared. And what we saw, if we remove any background noise introduced by differences in genetic background, that ES cells and iPS cells are very similar. But we saw a few genes that were expressed in ES cells but not in iPS cells. And to our surprise, they localized both to the imprinted DLK1 dio3 cluster. Specifically, there were non-coding transcripts normally expressed from the maternal, the unmethylated allele. In accordance with the absence of expression of these transcripts in iPS cells, the imprinting control region of this cluster, called the IgDMR, was hypermethylated in iPS cells, which we didn't see in control ES cells or in starting fibroblasts. If we knocked out the de novo methyltransferase DNMT3A in fibroblasts during reprogramming, we prevented the establishment of this hypermethylation. So this is the same enzyme that establishes this imprinting mark in male germ cells. So what's happening then during reprogramming is that the Yamanaka factors, together with DNMT3A, methylate the normally unmethylated locus control region of the maternal allele of DLK1 dio3, leading to epigenetically abnormal iPS cells that are also developmentally compromised. So we investigated this further, and we made by accident a very interesting observation. And that observation was that when we spiked in ascorbic acid into our reprogramming cultures during iPS cell formation, we very efficiently prevented DNA hypermethylation at this cluster. So you can see this here on the left. All of these orange bars, they are individual cell lines, and you can see that in presence of ascorbic acid, they maintain physiological degrees of DNA methylation, and these lines are generally developmentally fully competent. To the right, the yellow and blue lines show hypermethylation because they were derived in the presence of insufficient amount of ascorbic acid and are generally developmentally compromised. A side note, while ascorbic acid is very potent to prevent hypermethylation during reprogramming, it cannot reverse already established hypermethylation patterns at this cluster. So what can you do with this? You can, for example, now generate fully and faithfully reprogrammed iPS from difficult to reprogram cells such as B cells, and then use these iPS cells to make entire animals by injecting them into tetraploid blastocysts, which cannot give rise to the embryo proper themselves. So all of these two mice here, you see in the bottom, were entirely derived from a single B cell, and of course all of their cells have the same immunoglobulin rearrangement, including the immune system. So this is a nice proof of principle for the reprogramming of an adult cell type, but it also might have immunological study opportunities. So what we were really interested in was the realization that we had now in our hands a system in which we could study imprinting in culture. We could now, in a very reproducible manner, introduce an aberrant imprinting mark by reprogramming in absence of ascorbic acid, while maintaining physiological imprinting when reprogramming in presence of ascorbic acid. And applying this to a chromatin kinetic analysis of reprogramming intermediates derived under different conditions, we saw that what underlie this loss of imprinting was a rapid loss of active histone marks in absence of ascorbic acid that allowed transient binding of the de novo metal transferase DNA MMT3A exclusively in reprogramming intermediates in absence of ascorbic acid. So Emily Swansea, a student of mine, was very fascinated by using this imprinting instabilities to study mechanisms of epigenetic gene control. But she wanted to refine our analysis tools. And what she did, she knocked in reporter genes, different reporter genes into the paternal and maternal allele of DLK1. DLK1 is a protein coding gene that is normally expressed only from the methylated paternal allele of this gene cluster. So what she hoped that she would get cells that, if they were epigenetically healthy, express only the green fluorescent reporter, but if they acquire hypermethylation, they would now express also the red fluorescent reporter. So what did we learn from this system? First of all, it is a beautiful indicator for the stability of imprints in development. You can here see a mid-gestation embryo. You can see that the paternal allele is strongly expressed in many tissues of the embryo. The embryos are green. You can also see that that's the paternal allele. The maternal allele is hardly expressed at all. So this shows you that in development, imprint stability is maintained, and we saw a high degree of reproducibility here between embryos, hardly ever, actually never, any really convincing evidence of loss of imprinting. But in culture, this is of course different. And here the system allowed us to see really at high resolution the loss of imprinting that is associated with basal iPS cell formation. You see that here on the upper left. You see that all of the cells that express DLK are expressing DLK biallelically. So they are within this yellow gate. While iPS cells derived in presence of ascorbic acid almost exclusively give rise to cells within that green gate, largely paternal expression only. And this correlated very clearly on the right with DNA methylation, high degree of biallelic expression, high degree of DNA methylation, indicating that our system really captures the epigenetics underlying the regulation of this cluster. But we learned also a couple of new things. So for example, even in ascorbic acid-derived clones now, we saw a small amount of cells that appear to have lost their imprinting mark. And if you sort them out, we at least can test that they indeed have acquired DNA hypermethylation. So where are these cells coming from? So when Emily followed cultures over time, and that is now something that is true for induced pluripotent stem cells and for embryonic stem cells, she saw that even lines that started out as epigenetically normal at early passage, so early paternal expression, over time acquired more and more cells that had lost their imprinting. So there's an erosion of imprinting control in culture. And also very interestingly, if she looked at different lines derived from the same litter, there was a huge variation in the propensity to lose imprinting in culture. For example, if you look here at this ES10, it's extremely stable, while the brother line ES6 is very unstable. So there are yet to be defined intrinsic properties of these lines that predispose them to lose imprinting. So what that then means, essentially, there are two different ways in which your imprinting can be dysregulated in pluripotent cells. You can lose imprinting in a very sudden and abrupt event during iPS cell formation, or you can lose it over time in culture as a consequence of erosion. And of course, what we really want to know is what are the factors that drive this loss of imprinting, and what are the cell intrinsic factors that can counteract it? So what are the factors that try to preserve epigenetic integrity in pluripotent cells. And to get at that, what we're doing currently, and I'm not going to show you any data on that, we conduct a number of screens, different kind of screens to identify such factors. So to summarize this first part, the dysregulation of imprinted genes is one of the abnormalities that we find in pluripotent cells. I showed you this for mouse, that is true for human as well. Imprinting can be lost during reprogramming. That we can counter it very nicely for this cluster that I showed you by keeping ascorbic acid present during iPS cell formation. And it can also be lost during later culture of pluripotent cells. And while this is an issue for the use of cells, it is also an opportunity to study the molecular mechanism of epigenetic control at imprinted gene clusters and in general. So in the second part now, I would like to change gears. I would like to talk about the molecular features that characterize cells when they acquire developmental pluripotency during iPS cell reprogramming. And to make you understand what I'm really after here, is this really remarkable transition that reprogramming achieves. It turns a short-lived skin cell that has extremely restricted potential into an immortal pluripotent cell that can self-renew and has unrestricted developmental pluripotency, or almost unrestricted developmental pluripotency. So how is this achieved? And of course, over the year, we and many other labs have built roadmaps of iPS cell formation that described molecular, cellular, and transcriptional chromatin changes during the iPS cell formation process. And these roadmaps have been extremely powerful at dissecting the process further, but I'd argue that there's still a number of important black boxes here. And in particular, what we felt was missing is really an understanding when has sufficient molecular remodeling occurred for pluripotent cell features to actually be present in cells. Again, this sounds like a simple question, but it technically faces a number of challenges. One of the challenges I already spoke about, of course, if your reprogramming approach yields developmentally impaired cells, then there's not much sense in going back during reprogramming and ask when is pluripotency established. A second issue is the low efficiency of iPS cell formation. So even in genetically defined systems, it's always a minority of cells that reprograms after a couple of weeks. Meaning these successfully reprogramming cells that you really want to study and test are hidden in a failing biopopulation. That means if you want to do functional tests in particular, you have to first isolate those cells that reprogram and expand them. And at the point when you can do functional tests, you might be days or weeks after the initial acquisition of pluripotency, so you cannot learn anything anymore about this important phase of iPS cell formation. So Simon and a student in my lab and Bish, a postdoc, tried to increase the efficiency of our reprogrammable system. You remember this DOX-inducible polycistronic cassette that I mentioned to you earlier, by adding in a number of chemical compounds. And they screened a larger amount and ended up with 3 compounds that are called 3C compounds. The first one is a Wnt activator, the second is a TGF-beta inhibitor, and the third one is ascorbic acid, which we need here both to maintain epigenetic integrity, but also to facilitate the alteration of chromatin state, because ascorbic acid is a cofactor of DNA and histone demethylases. And what 3C does, it increases the efficiency of reprogramming of mouse skin cells dramatically from about 1% to over 50%, and it shortens the time that is required to reactivate important endogenous pluripotency loci such as OCT4. So if you put this all together, you end up with the reprogramming timing that is now less than a week, and the majority of cells make it at the end of the day. So that's very efficient, but we were worried that this high efficiency might come at a developmental cost, at a developmental impairment of the final iPS cells. And we were thinking that because of evidence in the literature that suggested that some of the slowest and least efficient protocols of making iPS cells yielded actually the best cells. Best cells as judged by the ability of these iPS cells to make entire mice when injected into blastocysts. So if there is really an inverse correlation between reprogramming speed and iPS cell quality, we should be in trouble with our system. But luckily that was not the case, quite the contrary. So iPS cells derived with our 3C approach, both from fibroblasts as shown here, but also from blood cells, were very capable of making entire animals and compared actually very favorably to other approaches described in the literature that are of course much less efficient at reprogramming. So what that means, we have now in our hands an approach that is not only fast, but that is also faithful. That means now it makes sense to go in at different stages of iPS cell formation and ask, are these cells able to self-renew as judged by clonal seeding? Are these cells pluripotent as judged by their ability to make mice upon blastocyst injection? And we did this at 3 different stages. The first stage are OCT4 positive cells that are still exposed to the reprogramming enhancing chemicals. Stage 2 is before the first time you passage your cells, but after you have withdrawn the reprogramming enhancing chemicals and doxycycline. So this is about a week after you start iPS cell formation. And the stage 3 is more what corresponds to your regular induced pluripotent stem cells after a couple of passaging in standard conditions. And what you see with self-renewal is that OCT4 positive cells at stage 1 are very bad at clonal cell seeding, but now then in a pattern that we kind of expected, you see a gradual increase in self-renewal potential that then peaks in established iPS cells. However, the kinetics of acquisition of developmental pluripotency are different. Again, S1 cells are doing very poorly, but now this potential peaks at the S2 stage. So these cells are extremely potent at making mice, and this potency even exceeds the potency of established iPS cells or established ES cells. So what that means that we have a kinetic here of pluripotency acquisition, so changes happening in OCT4 positive cells that rapidly endow them with developmental potency while the establishment on self-renewal lags behind. And the question is, of course, what is the molecular reason for these differences? And when you do RNA-seq, you cluster your samples, you see that you see evidence for reproducible molecular differences between S1 and S2 cells and established cells. So I'm going to talk first now about gene groups which are not different, which cannot explain the difference between pluripotent and pre-pluripotent cells. And the first difference is that the silencing of fibroblast genes is already completely achieved in S1 cells which are not developmentally pluripotent. Another group of genes that cannot distinguish are many core pluripotency regulators such as NANOG, ESRB, or OCT4 itself. These genes are expressed at very similar levels in S1 cells, S2 cells, and established iPS cells. And also another group of genes that can't distinguish are genes associated with this 2-cell stage embryo, which become activated only in established pluripotent cells. So what distinguishes pre-pluripotent and pluripotent cells? One group is a group of ectopic genes. So these are genes that are associated with somatic cells, but that are not found in fibroblasts. So for reasons that we don't understand, these genes become upregulated highly in S1 cells before becoming downregulated again. A second group of genes contains many germ cell regulators of multicellular cellular development, and these genes become upregulated in S2 cells and stay upregulated. And the third group of genes is a very peculiar group of genes because this gene group really spikes or peaks in S2 cells and then drops off again. And these genes are many transcriptional regulators of embryonic access formation. So I believe all of these groups warrant further investigation, but we were particularly interested in these transiently highly expressed genes. And one of the reasons why we thought these genes would be interesting is if you remember what I told you about the differences between naive and prime pluripotent cells. You see that a couple of these markers of the S2 cell stage are also markers of prime pluripotent cells, such as OTX2 and SIG2, 2 transcription factors. However, in contrast to our S2 cells, which are very good at incorporating into early embryos, prime pluripotent cells, of course, are very bad at doing so. So what is going on here? So we were nervous that maybe what we saw in RNA-seq would reflect heterogeneity of the S2 cell population. So we made a knock-in, inserting a reporter gene into the endogenous OTX2 locus. And to our relief, what we saw, this is in the upper left, that S2 cells almost homogeneously expressed OTX2, while only a small fraction, or like, well, a smaller fraction of established iPS cells expressed this gene. So are OTX2 positive cells in S2 cell, at the S2 cell stage and established cultures the same? They are not. So if you isolate them by low pressure cell sorting and inject them into blastocysts, you see that OTX2 positive cells at S2 make mice very efficiently, while OTX2 positive cells later on do not. So for some reason, this early reprogramming stages tolerate the expression of these primed markers. So what could be the explanation? I mean, one explanation, and that's a very high-level explanation, so to say, is that OTX2 and these other genes are incorporated in a transcriptional circuitry that's different. And if you look at, if you incorporate our expression data with published data set, you can see evidence for that. You can see in red our S2 cells, and they are different from established naive cells. Those are the embryonic stem cells in purple, but they're also different from established prime cells. Those are the epistem cells in green. So we don't know what OTX2 is doing in this stage, if it's doing anything, or if it's just a marker, but the consequences of the expression are very different than in other cell stages. So what I've told you in the second part then is that reprogramming protocols that are very efficient can yield fully developmentally competent iPS cells. With using such a system, we were able to identify cells that acquire pluripotency, and they actually have a unique combination of functional and molecular attributes, in particular a very pronounced developmental potential when injected into early stage embryos. And this potential is not impaired by the expression of prime pluripotency regulators in these cells. So then at the very end, zooming out again, what this all means for the use of pluripotent cells for biomedical applications. I've told you imprinting abnormalities is one of the abnormalities found in pluripotent cells, and what we really have to find and develop are systems that allow us to identify and eliminate factors that contribute to the occurrence of these abnormalities. And that would be, of course, also that also requires the existence of really informative assays that allow you to distinguish whether when a molecular feature is actually a bad feature or when it's a neutral feature. And second, data that I've shown in that second part suggests that it might be attractive for a certain biomedical application to use very early iPS cells, maybe even pre-passage, because these cells seem to have a very robust developmental potential, at least in certain assays. I've also shown you evidence that suggests to establish desirable naive pluripotency features, you don't have to erase some of the hallmarks of the prime pluripotent state. So with that, I would like to thank everybody in the lab that was involved in this work. I would like to thank our collaborators, in particular Effie Apostolous lab at Weill Cornell, Ruth Lehman, and everybody else at Scrabble, and of course funding sources. And I would like to thank you for your attention.
Thank you, Dr. Stadtfeld. As a reminder to our audience, you may ask questions at any time using the question box. Our panel will have an opportunity to respond after our next speaker. And that next speaker is Dr. Kejin Hu. Dr. Kejin Hu is an assistant professor in stem cell biology and cellular reprogramming at the University of Alabama at Birmingham School of Medicine. He completed his PhD program in molecular biology at the University of Hong Kong in 2003. Excuse me. He initiated his career in stem cell biology as a postdoctoral scientist at the University of Wisconsin-Madison in 2007. Since then, he has published more than 10 articles in cellular reprogramming and pluripotency. Dr. Hu is an ad hoc manuscript reviewer for more than 10 scientific journals, grant reviewer for multiple funding agencies, and is currently on the editorial board for the Journal of Stem Cells and Development. Dr. Hu's research is supported by the Alabama Institute of Medicine, the Faculty Development Fund of the current university, the American Heart Association, and the NIH. Dr. Hu?
Thank you, Catherine, for your nice introduction. Hello, everybody. I'm sorry, I have to say, today I have some allergy. If you hear me coughing, just forgive me. My research is cell reprogramming and pluripotency, especially human iPS reprogramming. Today, I'm going to share with you two stories in my lab. If you have any question at the end, you don't have a chance to ask. Here is my email. Just email me any question you may have. So, oh, my mouse is not very okay. Now it's working. So, today I'm going to share with you two short stories in my lab. But before I talk about iPS reprogramming, so it's a good idea to briefly refresh your memory about the basic of embryonic stem cell. When we talk about embryonic stem cell, we refer to the cell we culture in the lab, isolated from inner cell mass of a pre-implantation embryo. The first embryonic stem cell, embryonic stem cell 9 were established in 1981 by Martin Evans, Gail Martin, and Matthew Kaufman. It took us more than 17 years to make the same achievement in human. In 1998, Dr. James Thompson from the University of Wisconsin, he successfully cultured human embryonic stem cell. Human embryonic stem cells are invaluable resources because ES cell has two basic features that tissue stem cells do not have. First of all, embryonic stem cells have the potential to become any type of cell in our body. Secondly, we can culture embryonic stem cell in the lab forever. It has unlimited ability to expand. Because of these two features of embryonic stem cell, they have many critical applications. First of all, the greatest promise for embryonic stem cell is cell therapy. Also, embryonic stem cell can be used in drug screening, basic research, and disease modeling. Embryonic stem cells are infallible resource, but embryonic stem cells are not without any limitation. In fact, they have three critical limitations. First of all, not everybody likes the idea to use embryo in the research and clinical setting. Even though everybody is okay with using embryo in the research, we don't have sufficient supply of embryo. To obtain embryo is not like to get a cup of a Starbucks coffee on the street. Certainly, we have a critical technical problem in using embryonic stem cell in clinical setting. As you can see in this slide, to establish embryonic stem cell line, we have to use ICM, inner cell mass. But it is this ICM that developed to a human being. Using one embryo, we can establish a human embryonic stem cell line or naturally develop to a beautiful human being, but we cannot get both. In this case, this embryonic stem cell line is nobody's cell. So if you transplant the cell derived from this ES cell line into a patient, you will have a problem. You have immune rejection. To overcome this issue, scientists have looked for an alternative way to establish embryonic stem cell. In 2006, Dr. Shinya Yamanaka, the Japanese scientist, his lab discovered a new way to establish embryonic stem cell line. His lab successfully converted mouse fibroblasts to embryonic stem cell lines, neck cell. They named this cell induced pluripotent stem cell. Only 1 year later, his lab, using the same reprogramming factor OUT4, SOX2, CMYC, and KL4, successfully convert human fibroblasts to iPS cell. At the same time, Dr. James Thompson lab in Wisconsin also converted human fibroblasts into human iPS cell. They use a different set of reprogramming factor, OUT4, LANOG, and NIN28. Now we can convert a lot of different cells into iPS cell. Here are several examples. We can convert keratinocyte, T cell, B cell, blood progenitor cell, fibroblasts, and neuron stem cell into iPS cell. In fact, iPS cells have the same cell morphology, colony morphology, very similar transcriptome and epigenetic landscape as the ES cell have. Most importantly, iPS cells have the same differentiation potential as ES cells have. Like many other technology, iPS cells, the original technology, also have a lot of room to improve. Even now, in my opinion, iPS cells still have a lot of problem. It is still, this reprogramming is still inefficient. It is a process, slow, incomplete, stochastic. We have aberrant reprogram-associated mutagenesis and transformation. Today, I'm not going to discuss the detail about this improvement. If anybody are interested in the technical detail, I have 2 comprehensive reviews published in 2014. In these 2 reviews, I have discussed many technical details, but today I do want to mention one problem with the original protocol, because iPS reprogramming is a long process, especially for human iPS reprogramming. It takes more than 2 weeks. In this case, we have to integrate the reprogramming factor, these 4 reprogramming factor, into the genome to be reprogrammed. This integrating reprogramming has 3 critical problems. First of all, if you insert any foreign gene into the genome to be reprogrammed, you have a risk of insertion mutagenesis. Secondly, we know these 4 reprogramming factors are silenced upon completion of reprogramming, but much of the time, the silence is not complete. This incomplete silencing is indication of incomplete reprogramming. Also, all these 4 genes, the expression level of these 4 genes have to be in a narrow window. If you have a residual expression, your iPS cell will lose the identity. Finally, even all the transgenes are silenced completely, these genes can be reactivated anytime during the differentiation process or after you transplant this cell into the patient. To overcome this problem associated with the integrating reprogramming approach, James Thompson lab developed a new reprogramming system. It's called episomal reprogramming. These episomal plasmids do not integrate into the genome to be reprogrammed. This plasmid is very simple. It just needs 2 components from the virus. One is origin of replication is only 1.7 kb. The other is Epstein-Barr virus nuclear antigen 1. With these two components from the virus, this plasmid can replicate in human cell and also can partition when the cell divided. But this partition is not perfect. However, this is an advantage in terms of reprogramming. We can isolate iPS cell without when in the population lost the plasmid. Indeed, Kejin, my friend, she reprogrammed human fibroblasts into iPS cell and could able to isolate iPS cell without any transgene. We use different approach to prove this. We use RT-PCR. We don't see any expression of transgene. And we use genomic PCR. As you can see, there's no integration, no free plasmid. Our southern blot analysis also indicate there's no transgene in this iPS cell. So using this episomal system, we can human fibroblast into iPS cell. However, the expression, the efficiency of reprogramming is very, very low. When I was a postdoc in Dr. Igor Slopk's lab, we hypothesized progenitor cell might be easy to reprogram. So we use blood progenitor cell because blood progenitor cell are the most accessible progenitor cell in our body. I developed a protocol. I can reprogram blood sample, cord blood, and bone marrow frozen in the liquid nitrogen for more than 10 years. Indeed, as you can see here in this figure, episomal reprogramming of bone marrow is more than 100 times efficient compared to fibroblast reprogramming. Indeed, I isolated iPS without any transgene in this cell. Now this next slide this slide is a slide I made 7 years ago when I established my own lab. I still believe what this slide states still holds true. I still believe reprogramming is a black box to us because reprogramming even now is still very inefficient. It's a non-process. It is a multi-step process. It is still stochastic. We have reprogramming associated mutagenesis and transformation, and many cells are incomplete reprogramming. About this reprogramming black box, I have two major hypotheses then. First of all, I still believe there are additional reprogramming factors we don't know. And secondly, I believe there are a major reprogramming barrier we still don't know. Now I'm going to share two small stories. First, I tell you how we discover a new reprogramming factor, and then I will show with you how we overcome reprogramming barrier. As I said, I think there are new reprogramming factors we don't know. So to search for the new reprogramming factor, we bought a human Kejin CD library. Kejin Shao, my postdoc then, he evaluated the reprogramming activity of the human Kejin cDNA. After he evaluated 89 cDNA, he found some candidate, and then he did a secondary screening. He found one cDNA consistently enhanced iPS reprogramming, and then he tested this cDNA in 4-factor condition and 3-factor condition. Indeed, this cDNA enhanced reprogramming activity significantly in both conditions. Based on the library, this is a human BRD3 cDNA. We then purify this cDNA and sequence it. Surprisingly, this is a short isoform of BRD3. Because nobody studied this cDNA before, we named this protein BRD3R, standing for BRD3 with reprogramming activity. So we found a new reprogramming factor. So to understand how this protein enhances reprogramming, we conducted RNA sequencing using RNA from the only reprogramming cell. Interestingly, we found a lot of mitotic genes are up-regulated by BRD3R. You can see in this figure, we found a lot of geoterm contained the keyword mitosis, M phase, and related geoterm. Indeed, after we conducted more RNA sequencing, we found 128 mitotic genes are consistently upregulated by BRD3R. This indicates BRD3R has a mitotic activity. This is confirmed by our additional experiment. Our mitotic shakeoff experiment indicated that upon BRD3R expression, we have more mitotic gene. We have a similar result when we use flow cytometry to profile the cell cycle. Now, you know BRD3R has two activity, reprogramming activity and mitotic activity. Does it make sense? Can these two activity explain to each other? My answer is yes. I believe BRD3R mitotic activity is responsible for its reprogramming activity. To explain this to you, I have to introduce another reprogramming approach. This is outside reprogramming. Outside is in fact the most powerful reprogramming factor in the nature. Associated with outside reprogramming, we have another technology, somatic cell nuclear transfer. This cartoon explains to you how this technology works. You remove the nuclear of outside and then take a nuclear from a somatic cell. Most of the time is fibroblast. Then you put the fibroblast nuclear into the enucleated outside and let this reconstructed outside to develop. And then you get a blastocyst. Using this blastocyst, you can do two things. First, you can establish embryonic stem cell as you do using the normal blastocyst. This is called new NT-embryonic stem cell or NT-pluripotent cell, NT standing for nuclear transfer. Using this blastocyst, you can also to clone animal. The most famous cloned sheep, Donny, was cloned using this technology. Here I show to you this mouse is the first mouse cloned using somatic cell nuclear transfer. Her name is Cumulina. Using SCNT cloning technology, over half a century, we have two very interesting observations. For both recipient cell and donor nuclei, we know mitosis is very critical for cloning. In this table, you can see outside is a filter we use to clone. almost all the animal we clone now, but upon fertilization, zygote lost animal cloning capacity. However, Kejin from Harvard University, his lab did an experiment. His lab showed if you arrest zygote in mitosis, this zygote regained animal cloning capacity. Even for plasmid from a two-cell embryo, this plasmid has no capacity to clone animal. But Kevin's lab showed if you arrest plasmid from a two-cell embryo, this plasmid regain reprogramming activity. At the same time, John Gordon's lab, the Nobel laureate, in 2014, his lab published a paper. His lab showed if you arrest donor nuclei at mitosis, these nuclei are 100 times easier to be reprogrammed. Now you may understand why I said BRD3 mitotic activity may be responsible for its reprogramming activity. Now this concludes my first story. My lab screened a human Kejin library, and we discovered that BRD3R is a robust new reprogramming factor, and it might be that its mitotic activity contribute to its reprogramming activity. Now I'm going to shift the gear to another topic, reprogramming barrier. Talking about IPS reprogramming, many people say it is epigenetic reprogramming, it is cellular reprogramming. Today, I would like to use another term, transcriptional reprogramming, because if you want to convert one cell type to another cell type, what you have to do are two things. First of all, you have to establish the transcriptional program specific to the endpoint cell. At the same time, you have to erase the transcriptional program specific for the starting cell. Most of the time, it's fibroblast. Here, I have two heat maps to explain to you these two processes during IPS reprogramming. On the left side, there are a group of genes. These genes are embryonic stem cell specific genes not expressed in fibroblasts, not expressed in bone marrow. So if you want to convert fibroblasts to iPS cell, you have to activate this group of genes. At the same time, on the right side, this is a group of genes, Zolti fibroblast gene. These genes are expressed in fibroblasts, but not in iPS cell and not in ES cell. If you want to convert fibroblasts to iPS cell, you have shut down the expression of this Zolti gene. You may have experience to knock down one specific gene of your interest. You may encounter it's difficult to reach 80% knockdown. So you can imagine how difficult to shut down Zolti gene at the same time. Is this a whole picture? No. How big is the transcriptional barrier to IPS reprogramming? To answer this question, my lab conducted RNA sequencing to compare the expression of human fibroblasts and human embryonic stem cell. Here I show a heat map. As you can see, there are 3,148 genes that are expressed at a much higher level compared to human embryonic stem cell. Among these genes, 1,070 genes are not expressed over in human embryonic stem cell. What does this mean? This means If you want to convert human fibroblasts to human iPS cell, you have to shut down these 1,070 genes and downregulate the remaining more than 2,000 genes. This is a huge task. So how to downregulate these more than 3,000 genes efficiently and specifically? This is difficult to achieve, but today I will offer you a solution. But this is not a complete solution. To explain to you, what is our approach, I have to introduce to you a group of protein, bromine domain extraterminal family protein. Beta protein include 4 members, BRD2, BRD3, BRD4, and BRDT. Beta protein has a unique structural feature. They contain 2 bromine domains at their terminal. Bromine domain has a unique chemical capacity. This bromine domain combines acetylated histone. As you know, acetylated histone is an active transcriptional signal. So because of this feature, we call beta protein epigenetic reader. And also beta protein have another feature. This group of protein can remain associated with mitotic chromatin. I will explain a little bit more later. Because of this feature, we call this protein transcriptional bookmark. Now this slide shows you why we call beta protein epigenetic reader, because they bind to acetylated histone. As you can see, our confocal image shows that beta protein perfectly localized to acetylated histone. Here we use BRD3R and H3K9 acetylation as an example. But beta protein localized to a different loci from the heterochromatin marker HP1 alpha. Our histone pull-down experiment also show beta protein combined to acetylated histone, both for H3 and H4, especially for tetra acetylation. And this next slide show you why beta protein have another name, transcriptional bookmark. I'm not sure if everybody know. In any cell, when the cell are in mitosis, there's no transcription. They don't transcribe. This is called transcriptional shutdown, mitotic transcriptional shutdown. This is very clear if you look at the pore2 localization. Here is our confocal localization of pore2. As you can see, even in anaphase, pore2 dissociated from the chromatin. Because pore2 is not there, there's no transcription. However, beta protein remain associated with mitotic chromatin, as you can see here, from prophase to telophase. This is why we call beta protein transcriptional bookmark. Now you may understand our approach, because beta protein is an epigenetic reader and a transcriptional bookmark. And now the literature indicates that beta protein regulate cell type specific transcription and then define the cell identity. Based on this fact, you may have a natural hypothesis. That is, we can block the cell type specific transcription mediated by beta protein. Doing so, we can enhance iPS reprogramming. To do this, the best way may be to use chemical. Fortunately, in 2010, the bad family chemical is infallible. A Harvard lab, they report JK1 is a very good chemical inhibitor of bad bromine domain. As you can see here, JK1 perfectly fits the acetabidine packet. On this figure, you can also see JK1 have very high affinity to bromine domain, but bad bromine domain, but has no affinity or very, very weak affinity to other bromine domain. To test our hypothesis, we include bad chemical in our reprogramming cocktail. But this result, you can see, is disappointing. We don't see enhancement of reprogramming. In contrast, the reprogramming activity are impaired. We realize human fibroblasts express BRD2, BRD3, BRD4, and each of these proteins have multiple functions. And BRD4 play a very general role in transcription, that is transcriptional elongation. We just think the concentration we used in the experiment I presented to you in previous slide is a standard concentration in the literature. And we put the inhibitor in the reprogramming cocktail for 3 days. Considering this chemical inhibitor is not member-specific, we think the general function of bad protein may be compromised. And then we treat the reprogramming cell on day 3 and day 5. We remove the chemical on day 4. Interestingly, we found the reprogramming activity are increased even at high concentration, 1,000 nanomolar. Encouraged by this finding, we did a dosage test. We found if we use no concentration of chemical inhibitor, the reprogramming efficiency are significantly increased even for a long treatment, 11 days from day 3 to day 14 of reprogramming. And we found the optimal concentration is 50 nanomolar. This is 10 times lower than the widely used concentration in the literature. We also found at early stage, bad chemical inhibition have the highest reprogramming activity, but later treatment still benefit reprogramming. Also, continuous treatment for 11 days give us the highest reprogramming activity. We also found we use 2 additional bad chemical inhibitor. We have the similar result when we use them at a lower concentration. Interestingly, we found during reprogramming, bad chemical also change the cell morphology. As you can see here, fibroblast is now have a spindle shape, but in JK1 treatment, the cell become smaller, polygonal. Even some cell become round cell. This is very close to the shape of iPS cell. As I said, originally we hypothesized chemical inhibition can downregulate fibroblast transcription and enhance iPS reprogramming. To test this hypothesis, we conducted RNA sequencing. Indeed, when we treat the reprogramming cell for 48 hours at 50 nanomolar, we found 1,182 genes are downregulated. Of these, 651 gene have much higher expression in human fibroblasts compared to human embryonic stem cell, and 270 genes are not expressed at all in human embryonic stem cell. So this proved our hypothesis. We also found even in the non-reprogrammed cell, if we treat the human fibroblast for 48 hours, 551 genes are downregulated. Of these, 390 genes have high expression in human fibroblasts compared to human embryonic stem cell, and 162 genes are not expressed at all in human embryonic stem cell. Now, this concludes our second story. We find a chemical way to erase the fibrous transcription program. Doing so, we can enhance iPS reprogramming. Finally, I would like to thank my lab member, especially Zhichen Shao. He did a lot of work for these two projects. Rather, a good friend of mine, he did a lot of bioinformatics. Also, David contributed for some bioinformatics. Michael Crowney did the RNA sequencing. I also would like to take this opportunity to thank the scientists for offering me this platform to share with you my research. My research is supported by our university and also Alabama Institute of Medicine, American Heart Association, and NIH. Thank you so much for watching my seminar. I'm ready to take any of questions you may have.
Thank you, Dr. Hu. The audience has submitted several questions, so let's get to them. This is a question for both of you, Dr. Stadtfeld and Dr. Hu. You both mentioned the Yamanaka and Thompson factors used to create iPSCs. Is there much research into using different factors, and are these the ones that are always used, or are there ones that work better?
Do you want to go first? Should I go first? I don't care.
You can go first. You can go first.
I think one of the remarkable aspects of the initial factor combination that Yamanaka described is that it works so well. Okay. Efficiencies are low in some instances, but you will always get your colonies. And for some applications, you don't really care whether it's 1 in 1,000 or higher than that. No. So because it works so well, I think the factors were so successful that really most of the reprogramming that's ongoing is done with the 4 factors. Having said that, there are alternative combinations. I mentioned one of them described by Rudolf Jenish's lab in mouse fibroblasts that doesn't use any of the original like Yamanaka factors. Efficiencies are lower, but there seem also attractive features of this. Finally, reprogramming has even been reported entirely with chemicals without transcription factors. That could be also an interesting option. But if you look at what is used mostly and also like now for disease modeling and so on, it is the classical Yamanaka factors, still simply because they work well.
Yes. I think for both set of reprogramming factors, the efficiency are low, especially for human iPS reprogramming. That's why I look for new reprogramming factor. I believe still we have a very robust reprogramming factor need to be discovered.
Okay, great. Thank you for that answer. This is another question for both of you, I think. Recognizing the complexity of reprogramming, what are the current approved uses of stem cells for clinical therapy?
Do you want to go first?
You can go first.
So the clinical, I will answer this question for pluripotent cells, not for stem cells, because bone marrow transplantation, of course, is a stem cell like therapy that has been used for decades. Pluripotent stem cells, approved therapies. There are a couple of clinical trials that have been approved in Japan, but very recently to generate dopaminergic neurons in the brain of a patient. These are pioneering studies. There is no routine clinical intervention that is based on pluripotent stem cell therapy currently. But there has been, for example, another clinical trial that was medium successful to replace retina tissue in 2014, also in Japan. So there are some countries that aggressively pushing these cells into the clinic, but there is no routine process yet to use these cells for cellular regeneration. At least as far as I know.
Sorry, I have energy. Yes, as Dr. Stadtfeld said, iPS-based therapy is in the stage of clinical trial already. I have confidence in this kind of therapy, but in my opinion, now reprogramming, certainly we need a lot of improvement. We have to develop protocol for a GMP grade IPS for therapy. Also, as I said, there are a lot of a problem associated with current IPS reprogramming, associated mutagenesis, associated reprogramming associated mutagenesis, reprogramming associated transformation, and aberrant reprogramming, incomplete reprogramming. You can imagine for sequencing, in my time, my PhD program, I use manual sequencing, my big deal, but now sequencing, you have a next generation sequencing. For human genome, it took several years to complete, but now 1 week you can sequence the human genome. This is the case for IPS technology. I think for IPS to be used in clinical setting, we need to improve this technology.
I might want to add just one thing here, that there is also, of course, logistic issues with cell therapy using pluripotent cells. And that is simply the fact that you have to, of course, characterize your cells very well. So even if you have a perfect reprogramming protocol, you might have bad luck. You might have started with a bad cell. You might have had a mutation pre-existing. You have intrinsic instability of pluripotent cells even if they have never been reprogrammed. So you have to test these cells. And that actually is interesting because now the clinical trials that are ongoing in Japan, they're actually not using matched iPS cells for this reason. Because it's so difficult to actually, let's say, take a tissue sample from a given patient and then actually make iPS, test the iPS, expand the iPS, differentiate the iPS, and put them back into that same patient. So that is, you can do this as a proof of principle, yes, but to do this as a standard medical procedure is not attractive. So this logistic problem might really might be really the most serious problem for the establishment of cell therapy based on these cells. So I believe that the immediate potential is really in testing drugs, understanding disease. Because here you can discover general principles and find ways of interacting with disease features that are then applicable using drugs for many patients. And of course, that's so much more attractive for companies also than betting on cell therapy. So I am somewhat skeptical, to be honest, about the cell therapy perspectives, at least in the short run.
Okay, thank you. So you're sort of talking about there's still some logistical issues to overcome. We have another question here. Dr. Stadtfeld, I think this will be, we have just time for one more question. Dr. Stadtfeld, you mentioned that iPSCs can essentially be cultured forever. Do you know what's the longest time that iPSCs have been grown for, and have there been any issues?
Every time I make that claim on the slide, I swear to myself I will look into this question and be able to answer it. And actually, I don't know what is the longest. So it's a little bit of a bold statement then, forever. But having said that, even if you can certainly culture these cells for a year or so, and probably somebody has tried this, and I should know this. But there are issues, no? You have one feature of pluripotent cells is that they tolerate chromosomal abnormalities and other abnormalities much better than other cells. Other cells would start apoptosis, would start to die. Pluripotent cells don't. That is convenient, but it's also a big risk, because that means over time you can just accumulate abnormalities in these cells. And indeed, that's what you see. If you grow pluripotent cells for an extended period of time, they will acquire all kinds of abnormalities. Some are neutral, some are actually giving further growth advantage on top of the already impressive growth potential of these cells. I mean, maybe the most famous example is that the second X chromosome in female cells can be lost, in particular in mice. But there are other examples. So long-term culture is feasible, but your cells will drift into undesirable states on different levels, chromosomally, epigenetically.
Can I add something?
Yeah, sure.
Yeah. In terms of culture time, Dr. James Townsend, he established human embryonic stem cell. I have to mention his cell isolated in 1998. His cell line, first cell line, are still used worldwide in almost every lab in this area. Many cells are over 100 passage. Embryonic stem cell in the culture is not stable, but we can isolate a population that is normal. We don't have an experimental result to show how long we can culture this cell, but in theory, we can culture for a long time. Yeah.
Okay, great. So that was a very long time. Thank you. Unfortunately, that's all we have time for today. If you have any further questions, please consider reaching out to the speakers directly. Their emails are shown on the screen. As a reminder, the webinar will be archived on the scientist website, and you will receive an email notifying you when the on-demand webinar is available. I would like to thank everyone who took the time to join us today, and particularly those of you who shared your questions and comments. On behalf of the scientists, I'd also like to thank our speakers, Matthias Stadtfeld and Kejin, as well as our webinar sponsors, Biotechne, NanoString, Axol, and Stem Cell Technologies. Thanks everyone, and goodbye.